Okay, thank you all very much for joining our webinar tonight. This is another webinar in our gut-brain axis therapeutics series. I'm very pleased to introduce to you two very special speakers I've had the pleasure of collaborating with closely for many years. Dr. Rosa Krajmalnik-Brown is the director of the Biodesign Center for Health Through Microbiomes. She had the vision to found the center several years ago and has been seeing its rapid growth. She's a professor in the School of Sustainable Engineering and the Built Environment at Arizona State University. And she also is a co-founder of Gut-Brain Axis with me, and she serves as our Chief Science Officer. And we also have Nirmalkar, who's an assistant research scientist in the Biodesign Center, who's been working with Rosie and I for many years, and he's also the Chief Research Scientist for GBAT. So with that, I'm going to turn it over to my wonderful colleague, Dr. Rosie Krajmalnik-Brown, to tell us about some of the exciting research. Take it away, Rosie.
Thank you, Jim, and thanks for the invitation. So I'm going to start with our conflict of interest. That's how I call Khemlal Nirmalkar. Khem and I have several patents on FMT and Microbiota Transplant Therapy for autism and autism diagnostics. I am the co-founder and co-owner of two autism-related startup companies. GBAT is one of them, and the other one is Autism Diagnostics. And I have received some funding from Pinch Therapeutics and from GBAT for research, and Khem also has some equity in GBAT. So I'm going to start by telling you that the human gut microbiome is essential for many of the functions that we do and that we have in our body. The gut microbiome ferments our food to create vitamins and beneficial metabolites for us, and communicates with all our body, including through our immune system, through metabolites, and it also communicates with our brain, and it has some important functions also for metabolism. My research team has been working on learning a lot about the gut microbiome by creating or making interventions, clinical trials with interventions for human subjects. This way, we help people, and we also learn a lot about the gut microbiome. And some of the interventions that we looked at, we were the first team that looked at how bariatric surgery changes the gut microbiome. We've done a lot of work with diet and with probiotics, and what I'm going to talk to you about, which is something that has captured a lot of attention, is or work with microbiota transplant therapy for autism. Autism has a huge societal impact. The diagnosis of autism has been increasing dramatically in the last few years. The diagnosis in the year 2000 was 1 in 150 school-age children, and the latest diagnosis from the CDC that was published earlier this year is 1 in 31 school-age children. The diagnosis for ASD, Autism Spectrum Disorder, is based on behavior. There's no biochemical test for diagnosis, which is another huge need for the field. There is no cure or treatment available for ASD right now, and the estimated cost in the United States is about $461 billion per year. And so we look really closely at the microbiome, and I want to mention to you important factors that affect the gut microbiome, and they're all really nicely illustrated in this figure. So sex, the gender affects the gut microbiome. The diet is a very, very important component and determinant of the gut microbiome. The mode of delivery, C-section or vaginal delivery, physical activity, the environment, the host genetics, a few other health conditions, and any medications that we ingest also affect the gut microbiome. So some of you might already be thinking that some of the things that I already mentioned here are also things that have been implicated or correlated with increased diagnosis of ASD in the latest years. So keep in mind that all these things that have been also implicated in ASD diagnosis also affect and interact with the gut microbiome. One of the motivations for me to work on the gut microbiome and autism is, of course, my colleague Jim Adams, but he did a study in 2011, which was when he and I were already talking about collaborating, where he showed that gastrointestinal symptoms correlate with autism severity. And what you're looking at in here is a figure that on the y-axis, you can see a gastrointestinal score index. The higher the index, the more severe the gastrointestinal symptoms are. And on the x-axis, you can see autism treatment evaluation checklist. Again, the higher the number, the more severe the autism severity is. And you can see here a really nice line that shows this correlation between severity and gastrointestinal symptoms. He also showed me, when we were starting to talk about collaborating, that there was a study led by Dr. Sid Feingold, where they treated children with autism with vancomycin. And what they learned in this study was that in this figure, what you're looking at is the children at baseline before the treatment, during the treatment, and at follow-up. And what happened here, the higher the numbers, the better the behavior is improving. During the treatment, during vancomycin, the behavior of these children improved. But you can see here also that the follow-up, the behavior actually regressed. So these children were much better when they were taking the vancomycin, which is an antibiotic that goes all the way to the gut, that it's not transformed before it makes it to the gut. So it made it to the gut bacteria. And at follow-up, some of these or most of these benefits were lost. And so now we know a lot about the gut microbiome. And some of the things that we know, as I mentioned on my first slide, is that these beneficial microbes will modify bile acids, hopefully in a beneficial way. They produce butyrate, which is the food of our colonocytes. They produce vitamins. They produce folate, which is another important factor for gut microbiome and our brain health. And through some of these metabolites, also they interact with the brain and brain development. If we have an unhealthy gut microbiome, then we have a production of chemicals that are not exactly healthy, an unhealthy metabolite, and this can actually lead to neuroinflammation during development. And some of these metabolites also have been associated with anxiety and a few other important gut-brain connection disorders. So we started looking at the microbiome and autism more than 12 years ago. Jim and I have been working for many, many years together. And the first thing we did is look at, is the gut microbiota of kids with autism and typically developing kids different? And we looked at that, and many other people have done that. And what I can say in summary, number one, is that many studies have been done, different findings have been reported. In this paper that I put in here, which was meta-analysis that was published a year or two ago by our collaborator, Jamie Morton, he looked at many of these different studies. And one of the things that he found is that there's a lot of microbes that are found more abundantly present in kids with autism, more than 500. And there's a lot of microbes, about 150, 160, that are mostly more abundant in typically developing kids. But some of the trends that translate that are common to many of these studies and many of these papers is that in kids with autism, some of the things that people look at is increase of harmful microbes, and I put it in quotation, like Stutterella and Clostridium. And we were looking at this with a different class, and we were looking for perhaps a lack of beneficial microbes, and that's what we find, at least in our first studies. And so some of these beneficial microbes are Bifidobacterium, which is a typical probiotic, Prevotella, which is a microbe that is involved in fermentation, and it's also really important for gut health, and Lactobacillus. And also one of the things that we found is that most of these kids have lower diversity, like different, lower amount of different types of microbes, which is really important for a resilient gut. So in order to fix this, Jim and I are engineers, so we always think about how can we fix things? Can we fix it? We design an intervention, which is a microbiota transplant therapy. Initially, we called it transfer therapy, but now we've renamed it as microbiota transplant therapy. And with this therapy, initially we did an open label study with 18 children. And in this open label study, 18 children, we first treated the children 2 weeks of vancomycin. We emptied the gut. We provided a high dose of microbiota from a very healthy donor, and then 8 weeks of a lower dose of microbiota, which we call it the maintenance dose. And we collected a lot of fecal samples and blood samples and a lot of questionnaires for analysis. And the very first thing that I always want to mention about this trial is that it was open label, but amazingly, no one dropped out of the study, even though we actually ask a lot from our patients. So that's amazing in any clinical trial. So here are some of the highlight results of this microbiota transplant therapy study. So the first thing I'm showing you is symptoms, GI symptoms. So you're looking here at GSRS, which is a measurement of GI symptom severity on the y-axis. The higher the number, the worse the GI symptoms are. The lower the number, the better they are. Number 1 is actually pretty normal. So you can see here that our children to be treated started, this is our median, started right here, pretty severe. And at the end of treatment at week 10, a lot of these GI symptoms were actually much, much better. We had like an 80% improvement, which is amazing. Here when we designed our study, we designed a follow-up 8 weeks after treatment because we knew that on the vancomycin study, the kids lost their benefits after the treatment. So we wanted to see if there was any benefits. Do we lose them after time? And then later on, we did a 2 years follow-up. And on the 2 years follow-up, we can see that some of the children lost the benefits, but the average is much better than in the beginning of the treatment. So what are some of these symptoms that we saw improvement? Abdominal pain, diarrhea, and constipation. And so these are the GI symptoms. As far as the behavior, I'm showing you here a graph where on the y-axis is CARS, Child's Autism Rating Scale, and on the x-axis we have different timelines in the treatment. And you can see here with the lines, the delineation or separation between severe, mild to moderate, and minimal to no symptoms or diagnosis for ASD. And you can see that the majority of the children that participated in our study were in the severe category when we started the study. After 10 weeks, many of them dropped from the severe category to the mild to moderate, and some of them to even minimal or not being in the spectrum. At 8 weeks follow-up, some of them look even better. And when we followed up 2 years later, this was pretty amazing and surprising that many of these kids were either in the mild to moderate or in the minimal to no ASD, and only a few of them remained in the severe category. And I do want to point out that not everyone improved. Some of them did remain on the severe category, but the improvement was huge. So this is the behavior, and that's great. So what happened to the microbes? That's obviously one of the things that we are super interested in. I mentioned in one of my first microbiome slides that one of the things that was obvious to us was that the microbial diversity, which is how many different types of microbes are present in a gut, which is really important. One of the analogies that I usually give people to understand why the diversity is important, it's because we want a gut microbiome that it's like really diverse, like a society where you have many different engineers, lawyers, and doctors, and many of them. So if one of them gets sick, the other ones can do the job. So it's the same with microbes. A high diversity, it's always a higher and a healthier gut. So what you're looking in here in the y-axis is faith PD measurements, which is a measurement of diversity. The higher the number, the better. And then I want to guide you to this. The green line is 18 typically developing children that were age matched, that we use them as control. On the other red lines or orange lines are our donors. This is our initial donor, and this is our maintenance donor. And here's the median diversity of our children when they started the treatment. And you can see that when they started the treatment, they were lower than the typically developing kids, and then the donors that were providing the microbiota. 3 weeks later, we had provided a lot of vancomycin. So here there's not a huge improvement because they're still recovering from the vancomycin and just have also the inoculation of microbes. But at 10 weeks, we can see that the diversity improved. 8 weeks later, the diversity is improving even more. And when we came back 2 years later, the diversity is even higher than our typically developing kids, which this is amazing. This was very surprising and exciting to see how the diversity of the gut microbiome for these children was much higher. And not only the diversity was higher, but we were able to track also beneficial microbes. And so that, for example, in Bifidobacterium, which I'm showing on the very first graph in here, on the y-axis, we have a log of relative abundance. Here is the median, again, of the kids that we're treating, and the lines are the same. These are the typically developing kids and the donors. Bifidobacterium increased week 3, 10, which is the end of treatment, 8 weeks later, and even increased more at the 2-year follow-up. Prevotella, which is a microbe that we have been following, was almost depleted in most of the children. You can see 3D habit. Increased dramatically with treatment, 3 weeks, 10 weeks. And then it decreased a little bit at our follow-up points, but you can see that at 2 years after treatment is much higher than our typically developing kids. So this is a great outcome and great improvement. And one of my conclusions of this is that we changed the gut environment, and these kids were actually able to increase the diversity and the beneficial microbes, which is really important. I'm not going to talk about the sulfonylurea right now. But another thing that I can show you is that we wanted to see if the gut microbiome of these kids was similar to their donor. And this was, I forgot to mention, research done by a former postdoc that now is a professor in Toledo University, Daewoo Kang and Greg Caporaso, collaborators of ours. And in this figure, I'm showing you how similar, in general terms, is the gut microbiota of the kids that we treated to typically developing kids and to their donor. And so what you're looking at on the y-axis is an unweighted unifract factor comparison to our donors. And this is a measurement of this similarity. What that means is that the lower the number, the more similar it is to whatever you're comparing it to. So in the beginning, you can see here that the similarity of the kids that we're treating to their donor is about 0.5, 0.6, which is similar to the similarity that the average of the neurotypical kids have. So it's the same similarity as if you compare your gut microbiome to the person that would be sitting next to you at a conference. And so we see that after 3 weeks, this is much more similar to their donors, which is great. And here it's more similar to their donors than to the average of other kids, also 8 weeks later. But we can see that when we come back 2 years later, these children are not that similar to their donor anymore. They have a new beneficial gut microbiota. And so to look at this a little bit more into detail on what was the percentage of the donor and what was the percentage of their own microbiota that they retained, Chem did some amazing analysis on engraftment. And here we can see at least percentages of what was the amount of the microbiota that we see in our samples. Where did it come from? Did it come from the original microbiota or from the donors? And so I want to capture your attention first for 10 weeks, which is the end of treatment. And at the end of treatment, you can see here that about 48 to 49% actually of the microbiota that we measure was from the recipient, from the baseline or the recipient. And about 43 came from the donor, and there's like about 7% that came from unknown. We can call it environment, food. And when we follow up 2 years later, about 44% came from our recipients, and about 38% from the donors, and 17% from unknown sources. This gets me back to these kids now had a more beneficial microbiota, and also microbiota that was able to recruit beneficial microbes from food and from the environment. And I point out here that we can actually track the percentages of our Prevotella microbe in the end of treatment and after 2 years. And so we were successful in this open label treatment, but we as scientists want to know what are possible mechanisms of how this is working. And this slide summarizes what are possible mechanisms by which the gut microbiota interacts with the brain. And you can see here that some of the mechanisms are the immune system and the vagal nerves, but the hypothesis that we're pursuing is that bacterial molecules or metabolites are the factor that allows us to communicate from the gut to the brain. And so because we're looking at these metabolites, we leverage on the fact that we had fecal, urine, and blood samples for this treatment. And sorry about this. And here I'm showing you that when we look at the plasma metabolites of these treatments, one of the things that I want you to look at is this is a PKA analysis where each dot or square is the overall matrix of the metabolome that we find in each sample. And you can see on the squares, these are our typically developing children. This is how the metabolome looks for a typically developing sample. On the red circles, you can see the baseline kids, how the metabolome look in the children before we treated them. On the blue, you can see how it looked 3 weeks later, which in this case, we're really assessing the effect of vancomycin. And at 10 weeks, the green circles. And I want to call your attention to the fact that at 10 weeks, most of the circles are closer to our squares, which is the TD metabolome, the metabolome in the blood of the typically developing kids. So the metabolome changed dramatically from the initial baseline metabolome to be something more similar to our typically developing children. And this is something that it's really, really interesting because I can say here that what happens in the gut doesn't stay in the gut because our intervention was in the gut. And what ended up happening was that we can measure these levels of metabolites in the blood, how something that changed in the blood goes systemic into our individuals. And some of the metabolites that we measure that are important here, I have one that I want to show you as an example, which, sorry, this was changed in my order, is paracresol and paracresol sulfate. These are two metabolites that paracresol comes from tyrosine, which is an amino acid, and it's produced by microbes. And when paracresol can be changed by the host by sulfation and by some bacteria also to paracresol sulfate. And so we observe this paracresol sulfate in our samples at higher concentrations than in typically developing kids in fecal samples in the beginning of the treatment. And we were able to track and measure this metabolite during treatment. We can see that it goes down at week 3, and at week 18 is much lower, significantly lower, and much lower than even on our typically developing children. So this is a great improvement. And this paracresol sulfate has been correlated also paracresol and some of these metabolites that are metabolites of tyrosine also to other ASD-related studies with humans and with mice. And you might have heard Jim talk about our microbially derived metabolites, and this is one of them. So we also have a few other metabolites that have been important and mentioned for ASD that also were summarized in the meta-analysis that I mentioned before, where Jamie Morton looked at different studies and different metabolites and different microbes. And one of the key important things that has transpired in many studies with kids with autism and also with my studies are that important metabolites are microbially derived products of aromatic amino acids, tryptophan, phenylalanine, and tyrosine. And I just show you some important ones with tyrosine, which is paracresol. And the other important metabolite was glutamate. These are important because they are either precursors for neurotransmitters, like tryptophan is a precursor for serotonin, and tyrosine is a precursor of parathyroid, which affects some of the pathways for dopamine, for example, and glutamate also. And I was going to pass this to Cam, but I think I'm going to take it because we can't transfer the slides over, and I'm just going to take it and give him being the host in a second. And so this is work that Cam did with metagenomics, where he looked at techniques for sequencing that are more advanced than what I just showed you, where we sequence the whole genome of the microbes. And so we can get a better definition of microbes, and also we can look at important pathways that are represented or high represented or lower represented in all our samples. And so he looked at this metagenomics, which is genomics on a deeper level in our study. And one of the things that he found when we look at higher definition of microbes is similar to what I showed you before. Beneficial bacteria increase when we treated our kids with autism, and it became more similar to our typically developing children. But important things that he found also is that with treatment, we saw increase of genes that are involved in synthesis of folate, oxygen detoxification, which are really important, glutamate, which I just mentioned, and some important genes also that are involved in sulfur metabolism. So I mentioned paracresol gets sulfated. A lot of molecules that we ingest get sulfated in order to be able to detoxify them and get them out of our system. So these are really important genes also. And this correlated with lower autism and GI symptoms. So this is another study that I want to bring to your attention. So with this summarizing everything we did on our first phase 1 study for the microbes and the genomes of the microbes and the metabolites, I hope I convinced you with this that when we did this amazing treatment, the microbiome changed, but in the long term, each individual had a new personal beneficial microbiome, and that we were able to see systemic metabolite changes that show that changes in the microbiome affect overall metabolism. Like I said on my slide, what happens in the gut doesn't stay in the gut. That these GI improvements lasted for most, but not all participants, but they lasted for a long time. And that autism symptoms generally improve over the next 2 years in most, but not all participants. And I want to point out again that this was an open label trial. There is some placebo effect possible on the symptoms and on the GI, but not on the metabolites and not on the microbes measurements, because those are measurements that are done in the lab, and there's no placebo effect on those measurements, so they're very important. So with this, I'm gonna have to pause for a second because I need to make him a host so he can present on our phase 2 trials. 1 second. Okay, Cam, you're the host, so you should be able to turn on your camera and I'll try this.
Thank you,
Take it.
Rosie. Yes. Thank you. Thank you. So after completion of our open label phase 1 study, we conducted several phase 2 studies, which are listed here. Initially, we did 2 different phase 2 studies, one in adults and second one in kids with autism, and both were randomized double-blind placebo-controlled study. In a minute, I will share some of the results for the adult study. And the meanwhile, we conducted also another phase 2 study in children with the Pitt-Hopkins syndrome. Pitt-Hopkins syndrome is a rare genetic neurodevelopmental disorder where the individuals struggle with many severe autism-like behaviors. So we conducted this randomized double-blind placebo-controlled study. And since this is a rare disorder, we were able to enroll only 7 participants, but out of those 7, 6 were completed the trial. One couldn't swallow the pill, so was dropped out after starting the trial. But at the end of this trial, we saw that 80% reduction in the GI symptom in the treatment group compared to 30% in the placebo group. and amazingly 71% reduction in pain, and many other great improvement in the Pitt Hopkins symptoms. And this is, even though this was very small trial, but the results were very encouraging. And based on these findings, we submitted our results to FDA, and now we are requesting for the permission to apply for the drug approval. Currently, we are also doing two different other phase 2 studies, one with the larger cohort of the Pitt Hopkins syndrome individuals with our new formulated microbiota product, which is a powder version. With that, the individual won't struggle to swallow the pills, and they can take this powder version of the microbiota product with the dairy or non-dairy drinks. And also we are doing a autism dosing study, which to help to optimize the best dosage to get the best improvement in the autism symptoms and GI issues. So with that, we'll learn a little bit more about the adult study. Next, please. So, as I mentioned earlier, that adults with autism was a randomized double-blind placebo-controlled study where we enrolled 56 participants and where 51 completed the trial, which is a very high completion rate for the clinical trial. So this enrolled 56 participants were randomly assigned to two different groups, Group A with treatment and Group B with placebo. And this trial was in three different parts. In the part 1, Group A went with vancomycin, bowel cleanse, and 8 weeks of microbiota. Whereas in Group B, received a placebo pills and the bowel cleanse, because which we cannot hide. And the part 2, Group A, which is the treatment group, went for another 8 weeks of the microbiota treatment. But we wanted everyone to get treated. So Group B got a crossover, and they also started the treatment with the bowel cleanse and the 8 weeks of microbiota. Very important to note it here is that Group B did not receive vancomycin like Group A. The reason is our group and FDA wanted to know whether vancomycin is needed for the success of the trial and also the engraftment of some of the beneficial microbes that we are looking for. And the part 3 was an observation period until 18 months for the Group A and Group B both. Next, please. So the idea was that whether can we improve the symptoms of autism in adults using MTT treatment. Next, please. So here we are seeing a simple graph plot about how the autism symptoms changed. And Jim and his team worked very hard to see that how the symptoms are changing. So on the bottom part, which is a x-axis, shows the different timelines from the baseline until 18 months of the follow-up. On the y-axis, we are seeing many different number, which is for the CAR score, stand for Charlie Autism Rating Score scale for autism symptoms. And the higher, as it gets higher, the severity increases. And inside this plot, what we are seeing is that the gray color area, which shows the autism severity level, which is severe level of autism symptoms. And within this plot, also we are seeing two different colors. Orange color, which is stand for the placebo group, and the blue color is stand for the treatment group. And we can see here both of these groups, when they started this trial, they were in the severe autism level. And as they started the trial, at the end of part 1, we can see there are some changes happen. The symptoms a little bit of decrease, means the symptoms improve, we can say that. But we can see that treatment is improved a little bit better than placebo, but both are changing. So we can see a little bit of placebo effect as well. But we know that the treatment group got another 8 weeks of the microbiota treatment. So we can see much better improvement compared to the placebo group, because placebo group also got the treatment, but without vancomycin. So at the end, as we see here, the end of 18 months of follow-up, the treatment group improved much better compared to placebo group from the severe autism level to they reduce to mild to moderate level of autism for treatment group. Where in case of placebo group, they stayed in the severe level of autism. So what we are learning from this slide is there are three different things. First, the treatment groups improved much better than placebo at the end of part 1. But when they got a longer treatment, which is end of part 2, they showed much better improvement. So it means they need probably longer treatment. And the more importantly, that we need vancomycin most probably with microbiota to see the more improvement compared to the placebo. So that was three different takes that we needed to take, and that's why now we are running also the dosing studies to optimize that what dose would be the best to see the better changes in the symptoms. Next, please. So we saw the symptoms improved but we were also looking for what kind of microbes are changing. So here we're trying to show some individual microbes. On the left side, the blue color shows the Bifidobacterium species. So this is the average of many different species of Bifidobacterium. And Bifidobacterium is one of the most common beneficial bacteria that you can find in many different commercially available probiotics products or in your yogurt. And what we are seeing here in the green color plot, which is a box plot, each dot shows a abundance of Bifidobacterium species for each different individual for different timelines. From the on the x-axis, we are seeing from the baseline to and up part two when they finish the treatment, and the y-axis, which shows the abundance of this bacteria in the log scale. And the horizontal line, red and blue color, shows the average of the donors and the typically developing individuals. And what we are seeing here is that from the baseline, as they started the treatment, the abundance of this bacteria significantly increased. Next, please. But if we compare with the placebo, we did not see much change, which is on the purple color we are seeing here. But we know that placebo group also got crossover and they also got treated, which is end of part two. And we can see there was significant increase of this bacteria, which is in the green color here. Next, please. And similarly, we look for many different other bacteria, including the Bifidobacterium species, like Bifidobacterium longum here, which we are seeing on the right side. And it is similar kind of trend which we saw on the left side, that abundance of Bifidobacterium longum significantly increased in the treatment group after end of part one, end of part two. And which we did not see a similar kind of trend in the placebo group, which are in the purple group, until they got treated, which is in the green color at the end of part two. So that shows that the MTT not only improving the symptoms, but also increasing some of the beneficial microbes, including Bifidobacterium and Rhamnococcus and many other different that we haven't been able to show today. Next, please. So we learned in the two previous slides that the symptoms improve, individual bacterial abundance, which is a beneficial bacteria, also increase. Now we are seeing here that not only the important individual bacteria, but whole microbial composition also change, and they become very similar to donor. What does that mean is, when these individual adult with autism receive the microbiota product from the healthy donors, after the treatment, their microbial composition become very similar to their healthy donors. So let's understand this. This is a very complicated and colorful slide. So on the right side, what we are seeing is a scatter plot. We call here principal component analysis, principal coordination analysis for the placebo group. We are not seeing any specific kind of grouping pattern here, except on the right side bottom for the donor, which was from the University of Minnesota, which is in the dark green color. Now let's move towards the left side, which we are seeing for the plot for the treatment group. So this plot is trying to show how much they are similar to each other or dissimilar to each other. So let's focus on those are in the circle. We'll focus first starting with the red and orange color circle, which is a baseline group. And what we're seeing here, these are the individuals of each individual with their whole microbial composition before they started the trial. So first we know that they got a vancomycin treatment. Once they got treated, their whole microbial composition shifted away and they went far from the baseline. So we can see here, which we are seeing in the circle, light blue color and a little bit of yellowish. And once they started getting the treatment of microbiota, their microbial composition shifted towards their healthy donors. So in this study, we had two different kind of donors, University of Minnesota, which is in dark green color, and a black color, which shows the Finnish Therapeutics donor. So these two donors providers that we had, but once the individual of autism received the microbiota product, their composition our recipients composition shifted towards their corresponding donors, which we are seeing here in the two different directions. So that's a very powerful way to see that first, that the microbiota is working, MTT is working, and they're able to not only change increase the beneficial bacteria, but also changing their microbial composition as similar to healthy donors. Next, please. So overall, summarizing the results from the open label study which Dr. Rosie presented, and seeing the results from our adult study, that we can see that GI symptoms and autism symptoms significantly improve after MTT. On the microbiome side, we saw many different changes like Bifidobacterium, Prevotella, that significantly increase after MTT. We also learned that vancomycin pre-treatment was very important for the success of the trial to improve the symptoms, as well as with the engraftment of some of the beneficial microbes that Dr. Rosie presented, including Prevotella and Rosaburia. And about the metabolites, one of the potential harmful metabolite that we have been targeting for a while, which is a paracrisol or paracrisol sulfate. So in this case, paracrisol sulfate significantly reduced or significantly decreased after MTT. And more importantly, we saw that the plasma metabolite profile of autism with kids, they become very similar to TD after MTT. So overall, what we are learning from this overall the MTT trial for the specifically towards the microbiome, that MTT is helping and improving the GI and behavioral symptoms via modulating the gut microbiome and reducing of potential harmful metabolite that we saw in previous slides. Next, please. So with that, we would like to thank all those families and the participants who participate in these studies, and the donation from the many different families who help us to run this trial successfully. Our collaborator, University of Minnesota, and many different funding agency, including U.S. Department of Defense, Autism Research Institutes, ASU, and other all our autism research investigator and our group, and specifically Dr. Adams and Daewoo Kang, who was a part of the initial of these studies. And all of you, thank you so much for joining today and listening to us. Thank you.
Okay, thank you very much, Kim and Rosie, for the presentation. Just a few words from me before we start the Q&A. I want to mention that next week we also have a very special talk in the GBAT webinar series from a mother of a child with autism who observed great improvement in her son after receiving both ANRC and also from receiving testing by Anna Ludos, which then led her to do some additional treatments for her son. So that will be next week. And then also, next slide. I also want to invite you to consider investing in GBAT for those of you who are interested in helping us continue this type of research. GBAT is a very unusual company funded by over 120 autism families who've chosen to invest to purchase stock in our company. If you can forward the slide next. So these 120 families have been committed to making a difference in their child's life and the lives of millions of others by funding our research so we can do the next steps. We're still in a fundraising round. We need to raise about $20 million to be able to do our final phase 3 studies. So the more money we raise, the sooner we'll be able to move towards our final study for FDA approval for this treatment for autism. Next slide. Also, the investors have the potential for very large returns because pharma companies can be very, very profitable. We've had 4 successful previous trials to substantially reduce any investment risk, and our next 2 trials are still going very well. And we've got a great team of researchers working with us with over 10 years of experience. Next slide. And so if you're interested in learning more about investing in GBAT, please consider attending one of our next investor meetings. Those will be on Tuesday, December 16th and Wednesday, December 17th. If you want to learn more about gut-brain axis, you can also go to our website, and you can also email me at james@gutbrainaxistherapeutics.com. So with that, we're going to open it up for the Q&A. And so let's go ahead and turn on the chat so you can begin typing messages into chat. So Rosie is the host, if you want to turn that on.
Kim is the host now.
Kim, if you want to go down to options and then turn on chat.
Should I stop sharing the slides? I don't know that I can see the chat if I'm seeing the slides, Jane.
It's up to you. I think it's useful to show the slides for some of the questions.
No, I know, I just can't see the questions, just letting you know.
Rosie, can you try to type something? Because it looks like
I can't see the chat. Because I either see the slides or the chat. I can't see the slide. Oh, hold on. Maybe. Okay, I have it here outside. Try to type something. I can't type anything. It's closed. The chat is closed. It said chat disabled.
Okay, Kim, do you want to?
Now it's enabled.
Okay.
I think I can type something.
So everyone, please go ahead and type your questions into chat. I'll also mention that this meeting is being recorded, And so we will send a copy of the recording to all of you and everyone else who is registered for the meeting. And that should be available in a few days.
Okay, I see a lot of highs, but no questions yet in the chat. I can see it now. Jim, they want you to type something. Hold on.
Okay.
Oh, okay.
So I think we can see some of the questions. We are seeing one of the questions says, can you give your opinion about leucovorin? Jim, do you want to take this question?
Sure. So leucovorin is a high dose B vitamin. It's been investigated for many years. We've used a modestly high B dose vitamin in our vitamin mineral supplement for over 25 years now. There's many decades of research showing that high dose B vitamins can be helpful to children with autism, but in particular, leucovorin is an extremely high dose of a special form of folate. Folic acid is the artificial form that's commonly used, but that seems to not be well metabolized by children with autism and actually may be a problem if you give too much of it. Whereas the form of folate in leucovorin, folinic acid, is a very useful form. So I'm actually the chair of the Data Safety Board for 3 leucovorin studies run by my colleague, Dr. Richard Fry, who's really been pioneering the use of leucovorin for autism. The bottom line is that it does seem to be safe in most cases, a few rare cases of some increased hyperactivity, but in general, very safe and seems effective. Seems to be more effective if people have antibodies against the folate transporter that transports folate into the brain. And so that's a quick summary of it. That's not the focus of our meeting. But let's go ahead with other questions.
We have another question says, are you only eligible for studies in Arizona or elsewhere? So most of the studies are nationwide. We have been doing within the United States. Rosie and Jim, do you want to add anything else here?
And our future studies will also be nationwide.
Yeah.
Yes.
The next question is, are you still doing urine analysis? Yes. Jim is doing a lot of these microbially derived metabolites measurements in urine. And I assume that's what you're asking about, the urine analysis. So we do urine analysis in all our trials for research purposes. But now if people are interested in doing that urine analysis with their children, maybe Jim can put the information on the chat. This can be done. This microbially derived metabolites analysis can be done, and I think it's a good idea.
I'll put that in the chat.
Do you find similar success with bifida infantis? It says bifida implants. I don't know if you meant bifida infantis or.
Probably I think that's their meaning.
Okay, you want to answer, Cam? You want to take it?
Yeah, so we saw like changes in many different bifidobacterial species, but infantis is for the kids. Mostly they are in the kids. But the other study that we show was we saw adolescentis, not infantis. We are still working on the analysis for the kids with the autism, and there are which probably we may need to look into that. But so far we haven't seen for the infantis significant change. Yes, but they are there. Yeah.
The next question says, is there any way to have my son receive this treatment other than being in the clinical trials at this point? The answer, unfortunately, is no, not at this point. That's why we're working so hard to trying to get FDA approval, because that's the only way that a lot of families would have access to this treatment if we get FDA approval. So right now, the only option is the clinical trials. I don't know if you want to add to that, Jim.
No, I think you said it well. That's why we're trying to raise money to do those final phase 3 studies to get it approved by the FDA and available to everyone.
The next question says, is there any data about MTT via oral route versus endoscopic route? On our first open label trial, on the first inoculation, we did oral versus, I don't know how to call it. And it wasn't rectal.
Rectal enema.
It wasn't endoscopy, but it was rectal. The sample size is small, so we can't do exactly statistics to separate it, but right now, most of the trials we're doing are oral, not endoscopic. Which probiotics were given and for how long and how often? This is not about probiotics. This is a microbiota transplant therapy. The microbes are not probiotics. I mean, you can call them probiotics, but they come from very healthy humans. So it's microbiota that is purified from fecal human samples. And I mentioned how often they were given depending on the trial that you're asking the question about. In our first study, we did 8 weeks for the children. With the adults, Chem showed you can answer, Chem, about the adults.
So adults also, like, we did that treatment of 8 weeks of microbiota and then so the treatment group got actually twice, so almost we can say 16 weeks of microbiota, where in case of the placebo group only 8 weeks without vancomycin. So it depends on the what trial that we are talking about, yes.
It might help to mention the very high number of bacterial species present in the MTT product.
Yeah.
So that's one of the big advantages of the MTT product over just one of them over probiotic and high diversity, a huge amount of microbes, like about maybe average of 500 per, I don't know how you call it, per capsule.
500 species per capsule.
Yeah, per dose per capsule. When you buy probiotics on the store, maybe you find 12, 15 species on them, if you're lucky. Really high amount. And also these are microbes that their original ecosystem was a human gut. When you purchase probiotics on the store, most of them are microbes that their original ecosystem was milk, not a human host. So our microbes have a higher chance of surviving and thriving because of that. How can we find out about possibly being in the trials? I don't know, Jim, if you want to add something to the chat so people can email you and sign in for future trials.
They can sign up for our email list at autism.asu.edu to get on our general email list, and we'll notify them about future studies as soon as we have the funding for them.
When will the next MTT study be done on autism with level 3, on adults with level 3 autism? I'll give you that question, Jim.
Sorry, I didn't quite catch that. Can you say it again?
When will the next MTT study be done with adults?
We hope it would be within a year if we can raise the 20 million that we need for our next study. So we're working very hard at that.
Okay, this is one for you. It starts with Jim, so I don't know if you want to read it or you want me to read it for you.
I'm not sure where you are, so if you can read it, that would be great.
Jim, I have been tested for allergies and I tested for H+ on literally everything. So how much does MTT improve allergies?
It's a great question. If you come next week, you'll hear about a boy who had extreme allergies. He carried Kleenex with him every day, had terrible allergies. And when his gut was treated, not by MTT, but through the other treatments, he now is allergy-free. We had one child in our MTT study who had severe life-threatening anaphylactic shock reaction to nuts. And after treatment, he's now no longer has that life-threatening allergy. So the immune system is very tightly tied to the microbiome. There are studies now at Harvard Medical School with the product from University of Minnesota for treating peanut allergies, people with life-threatening peanut allergies. So it's amazing how much microbiota transplant can do.
Can this procedure be beneficial for a 42-year-old male who has had gastrointestinal problems for the past 20 years? I'm assuming this is an adult with ASD. If that's the question, I'm assuming the answer is yes. What do you think, Jim?
We treated people 20, 30, 40, 50 plus years, adults with autism, and they were able to see substantial improvements in their gut symptoms. So we think in most cases, yes.
Is the next study strictly for Americans?
They have to be living in the United States.
So you don't have to be American, but you have to be living here. When are you planning to do the phase 2 trial, and how many participants are you planning to enroll? Will there be adults in the study too?
We hope to enroll 200 people is our plan. We hope the FDA will let us enroll half children and half adults.
Yeah, but we depend on FDA recommendations.
That's true.
Can you please give your opinion on glutathione? It's an antioxidant, and it's a detoxifying molecule. But I don't know if Chem or Jim want to say more on glutathione.
I'll let Jim speak because he's been focusing on one of these metabolites also.
Glutathione is very important. Many studies show children with autism have lower amounts of glutathione, and they have trouble recycling the glutathione from the inactive form, the oxidized form, to the active form. It's the most important antioxidant in the body. It's also important for detoxification of toxic metals and many other things. We found from our vitamin mineral study that we were able to greatly improve both the level and the amount of active glutathione. It's a large molecule, so it doesn't seem to be very effective to give it by itself, but to instead give the precursors to make it.
My son is exclusively G2 fed. Are you planning on studying other options for delivery in the future?
We are treating people now with Pitt Hopkins who are fed by G2, so that's not a problem.
Did you keep the participants on the same diet? That's a great question.
Yeah.
Yeah, that's a great question. We asked them not to change their diet. So the idea was to keep them on the same diet. And on our first study, on the open label study, we monitored the diet in the beginning, but not later on. On the studies that we're running right now, we're checking for diet initially and also later on, because there is a possibility that after the treatment, these children get more comfortable eating other things, and that might be one of the reasons why their microbiome diversity increases dramatically too. So that's a great question and a really important one. What was the sample size for the MTT study? For the first one, 18 kids. For the adult study, we shoot for 56, right, Kim?
Yeah, and the 51 completed the study. Yeah. And around 50 also for the kids with autism. Yeah, so it depends on the depends on the study, but now we are focusing at least 50 so far, like in this 2 study.
Yeah.
Yeah.
So Jim, we're closing the hour and the list keeps on growing. So I don't know how long do you want to do this, or what's your plan for this?
I would suggest another 15 minutes and
Okay.
then invite people to email us questions later.
Sounds good. All right. I'm just asking since you're the boss here right now. Okay. Hi, thank you so much for this great information and for the work you do. It means the world. 2 questions. Have you seen a difference with the age of the patient? The older, the harder it is to get better. And the other one, when do you estimate that the treatment could be commercially available for the public? So to answer the first one, initially on our first study, we didn't see a difference with age, which is why we were very encouraged to do the adult study. So there wasn't a big difference on our first study with age. I'll let Jim and Chem add if they want to.
I think that's a great way to answer, Rosie. For the adults also, we as far as for the microbiome, we haven't like checked the very age wise. There is some age like we have from until like what you mentioned around 50, close to the 50 years old until the from the adulthood starts. So we have a big age range, but we haven't checked for the microbiome part yet. But we have overall, when we consider as a covariate the age, we haven't seen any much difference. For the symptoms, Jim, do you have any things to comment on that?
In general, I would say our first child study showed more benefit. The
Yeah.
children with severe autism went down mostly to mild autism or below the cutoff for mild. With the adults, they went from severe down to mild to moderate. So still substantial improvement, but perhaps somewhat less for adults. But it's hard to compare the different studies.
It's
Um,
also hard to compare because the donors were different, Jim, and this is something that we haven't deeply studied yet.
right.
Yeah.
And the
And so
second part of the question.
the second part of the question was, when do you estimate that the treatment could be commercially available for the public? We're working hard on that. So we need to run a phase 3 trial and get FDA approval, and then it can be commercially available for the public. And we're hoping that's soon. We're working very hard on that. I don't know if you want to add anything to that, Jim.
I'd say 3 to 5 years, depending on how long it takes us to raise the funding for a final phase 3 study.
Yeah.
Yeah.
Have you used the same healthy donor for all studies? Have you seen a difference if the donor is different? That's a great question. No, we didn't use the same healthy donor for our phase 1 and our phase 2 adult study. And I mean, Chem actually showed how we had 2 different batches of donors for the adult study. We did use the same donor for the some of the adult study and a phase 2 child study that we didn't show the results today. The donor is a great question. That is something that we want to investigate further also. If there are better donors or better matches between the donor and the recipient is a really good scientific question that we need to investigate further. You want
Good
to add something,
questions.
Cam?
I think you said it enough, Rosie, because this is something we are like keep like thinking about and how we can improve. So that's really great questions. Yeah.
You mentioned a few species which abundance was increased after MTT, but I'm curious, are you also investigating which species might have decreased in abundance in response to treatment? That is also a great question. Do you want to take it, Cam?
I'm just going to read one more time. You mentioned a few species whose abundance was increased after MTT, but I'm curious, are you also investigating which species maybe decrease abundance in response to treatment? That's a really great question. We have seen changes in some of the abundance of the bacteria that decrease specifically. For example, for the Pete Hopkins, we saw that some of the Clostridium species actually significantly reduced, even though it's a very small sample size study. So we are still working on that to figure to see that what are the other common bacteria that are decreasing, or sorry, other potential harmful probably. But Clostridium, few species that we know that they are potentially harmful based on the previous literatures from the Pitt Hopkins study and another different study. That's something we have seen that.
Maybe I could just add that there seem to be many different potentially harmful species,
Yeah.
but so there's very wide range from one child to another. But what we see is a common increase in the beneficial ones. So I think Rosie's been really powerful in saying the real focus is let's try to increase the beneficial ones.
Yeah, this is one of the things that distinguish us from other groups since the very beginning, that we were looking, and maybe it's a little bit of my fault because I'm an environmental engineer by training that loves microbes and that knows that microbes can do a lot of good things for us. And so we were looking at this with different classes of like, are we missing beneficial microbes? Are these kids missing beneficial microbes? And luckily, the answer was yes, because that's kind of like an easier thing to fix. Let's add the beneficial microbes. Let's restore the microbial ecosystem. And that's what we've been doing. And so the beneficial microbes in your gut also protect your gut, protect the lining, and also fight off the non-beneficial microbes. So if you can just increase the beneficial microbes and provide a better ecosystem, everything will be better. So, the non-beneficial microbes usually live in our intestines at low concentrations and low abundances, and they're fine. They're okay there as long as they're under control. Clostridium difficile, which is one of the bacteria that creates this lethal infection, most of us have some of that in our gut, but it's under control. The problem is when they take over and they actually take over the system and don't, they increase. So as long as we have a good amount of beneficial microbes and a happy environment for our microbes, things are usually good. Okay, Christina actually said, thank you, Professor Adams, me, and Kem for our work and for our presentation. And she asked, what is the most valuable lesson you've learned in your work on ASD? And we've learned a lot of them. I can tell you mine, and then I'll pass it to Kem and to Jim. Mine is what I just said, that, that beneficial microbes are really important. And if you can restore the ecosystem and add good microbes and beneficial microbes, you can actually change the gut environment, and you can change not just the microbes and how they interact with each other, but how they communicate and the chemicals that they produce. And that's really important. That's really important for the host, like to have butyrate produced, vitamins produced, and all this good stuff that microbes do for us instead of them producing metabolites that are toxic or that hinder the production of beneficial neurotransmitters also. I'll pass it to Kem and Jim and see what they can say about their, what's the most valuable lesson that they've learned.
Interesting question. Yeah, Rosie, I followed the same similar kind of answer because microbiomes are very resilient. It's very hard to distinguish and follow one specific microbes because the cohort, genetics, food, environment, many things matters. So it's very hard to distinguish which one is doing good or bad, specifically if the cohort size are different, the age and everything's are different. But more importantly, that what I am try to learn more is that microbes they are changing, but more importantly why they are changing, what kind of functions they are doing. So the functions are important, what kind of genes they have, how they are expressing themselves, are they producing specific kind of beneficial metabolites or molecules, or the harmful ones, are the one which are actually responsible for this kind of behavioral changes or GI issues. So that's something I'm still working and try to learn more about that. So microbes are resilient, and it's very hard to understand that, but we're trying very hard to understand based on their function, based on their function.
Jim, do you want to add, or should we move to the next question?
Let's just move on.
Okay, that's good. How can I endorse my child for an MTT trial? My child has H. pylori and candidas. I think you just mentioned that, right, Kim? How can they email you to get on the list?
Yeah.
Yes. And for candidiasis, again, there are antifungals available now they could be using.
Yeah, and we also have information on fungi, what we didn't present it today. We've been following also how the fungi changes also with MTT. Candida is a fungi. That's why I mentioned fungi, sorry. My son, who is 21 now, he was part of initial trial, but he got COVID during the trial. We also saw him show a diff, and he lost it. He lost it. We spoke about his need of high dose as he was grown. Can he participate in the adult trial now in figuring the dose? That's for you, Jim.
Yes, people can reapply.
Okay. Bifidobacteria implants through enema. I don't know. That's not a question. It just says here. Thank you for the work you're doing. Are you analyzing both donor age and recipient age in your MTT studies? Are you also tracking additional patient outcomes such as weight change or disease-related risk factors? Also, are typically developing siblings of children with ASD being considered as potential donors? I'll let you take that, Jim.
Okay. So for donor age, we are looking at many factors in the donor. For Clostridium difficile infection, almost any healthy person can be a donor. But for autism, it really matters who the donors are. So we pick the top few percent very healthiest people. So we think that's really the main factor. Adults have a higher diversity of bacteria than children, which is why we use adult donors. We are tracking many patient outcomes, including weight change. We have seen some children with Pitt-Hopkins who had very poor weights. They were very delayed, put on a healthy weight. And we are generally not considering typically developing siblings of children with autism as potential donors because siblings often have many of the characteristics. They're often halfway between the typically developing children and those with autism. Rosie, back to you.
Okay. How long does a fecal transplant remain effective? My son had improvement 3 years back, but now he's regressing. We don't know. We just know that 2 years later, kids were doing much better. And this is the first time I hear about this. I don't know if you knew any more about this, Jim.
We've had reports of people showing benefit up to 5 years. Antibiotics seem to be the major problem. Use of antibiotics can kill off many of the bacteria we're producing. Also, it's important to feed the bacteria. And Rosa, maybe you want to talk more about the importance of healthy diet.
Yeah, but fiber. High fiber helps you feed your beneficial bacteria. So high fiber, non-resistant starch, non-processed food. Those are some of the things that help you feed your beneficial bacteria, and it's very important. And so hopefully the children can do that after treatment. This goes together with what you just said, Jim. It says, have you heard from any of the participants past 2 years? Were there any more improvements? I think you've heard from some of them, right, Jim?
Yes. I would say that over the 2 years, we've heard the most comments that children just seem better able to learn, and hence were better able to begin catching up to their peers in terms of language, in terms of social interaction, in terms of better behavior. So in most cases,
yeah, some of them went to college. I know that some of them, at least a couple of them, went to ASU. Sorry, Jim.
Yes, absolutely.
Do you have any plans for additional longitudinal study follow-ups past 2 years, possibly into adulthood? Even the normal follow-up we do right now, it's a little bit challenging. So I'll let Jim comment on this, but it is a little bit challenging to follow up farther than that.
It's burdensome to families. And so when we enroll them in a study, we have to tell them how long the study will involve. And after children are doing well, families are less interested in following up in studies. So at the moment, no, we're doing shorter follow-ups. In the past, we have done, as I said, up to 2-year follow-up.
Yeah. Did you say the microbes are oral in your trial? Yes, they are oral in our trial. My son takes leucocorin 10 milligrams daily. Is that high enough? I am not an MD, so I'll let Jim answer.
A typical dosing is 1 to 2 milligrams per kilogram body weight. So it depends on what a person's diet is. There's still studies to find out what is optimal. The nice thing about B vitamins is that they're generally very safe because they're water soluble. So if you take high amounts, you just pee them out in your urine. So there's still studies being done to see is even higher dose beneficial for some people. So I think the best way to answer it is to try a higher dose and see if you see more benefit.
Yeah. So it's 6:45, Jim. Do we want to take
I think we should
by email?
I think we should thank our two speakers very much for their wonderful presentation. The recording will be sent around, and if you have additional questions, you have Dr. Rosie's email address here, and also I put mine into the chat. So many, many thanks for the wonderful research you two have been doing. It's been a pleasure to work with you for 15 years.
Thank you so much, Jim, for inviting us, and thank you everyone for joining us today.
Yeah, thank you.